Hello, learners. How are you? I hope you would be fine and be listening my videos continuously. Those learners, in the previous videos, we have discussed about asexual reproduction, sexual reproduction, the whole reproduction process we have discussed. But today we are with a new topic, that is heredity and evolution. There will be number of videos regarding this topic, and today we will discuss about the basics of heredity. So students, we resemble our grandparents, our parents, our siblings, our most of the relatives, maybe maternal, maybe paternal. We resemble most of our relatives. Why is it so? Maybe some information would be carried from one generation to another. Yes, students, this is correct. This information, the carrying of information from one generation to another is the study of genetics. We study about the genetics, genes. Now, these genes are the carrier of this genetic information which goes from one generation to the another generation. And student, evolution is one of the term. What the evolution? It is a branch of biology that deals with the changes that it causes in the diversity of living organisms over the period of time. So there are some evolutions also, there are some diversities. We totally don't resemble our parents, grandparents, paternal siblings. We don't resemble. There are some diversities also in our characters. So this leads to an evolution of another generation. Okay. The similarities and differences among the members of a species are not coincidental, children. They are received by the young ones from their parents. The hereditary information, in fact, is present in the gametes. Gametes we studied, these are eggs and sperm. They carry the hereditary information, which fuse to form the fertilized egg or zygote during sexual reproduction. The zygote then develops into an organism of a particular type. For instance, eggs of sparrows hatch into a sparrow only. Eggs of fertilized eggs of a pigeon hatches into pigeons only. The offsprings will be similar to their guardians, isn't it? Similarly, a cow gives birth to cow, buffalo gives birth to buffalo, giraffe gives birth to giraffe, and elephants to elephants. There is no change of a species while giving birth. So some of the genes are responsible for carrying the similar information, similar characteristics from one generation to another. So what is heredity? Heredity is a transmission of characters from one parent to the offsprings. That is from one generation to the next generation. It is called heredity. Now, some variations also occurs. Not every and each information results in the production of a similar offspring, isn't it? So what are these variations? This is the difference in the traits shown by the individual of a species and also by the offsprings of the same parents. It is referred as a variation. Okay, these are the changes. Okay, that we not a clone of our parents. So these changes are the very brief. Genetics is a branch of biology that deals with the study of hereditary and variations. The term genetics was coined by William Batson in 1906. So this was all introduction of about what is heredity, what is genetics, and what an evolution and variations are. Now, how the accumulation of variation occurs. So, heredity involves the inheritance of basic body design, like similarities which we carry from our parents. But there are subtle changes, some variations from one generation to the next generation. That is, when parents consider an offspring, then this individual has some its different characters. The second generation will be having a basic body design. It is means a human species. It has particular traits which a human species carries, but it is not just a copy of that. Then why is it so? These are the differences. While this is what we are discussing is about the sexual reproduction, isn't it? But What happens in asexual reproduction? Asexual reproduction has a single parent, and this single parent reproduces asexually, and this reproduces to form first individuals, then it forms a 4 individuals. In this way, large number of individuals are formed. After many generations, all these individuals would be similar. Why? Because there will be a very minor difference, or they will be exactly similar, because the thing is that they are from one parent. And the differences are also because there must be some difference in DNA copying. DNA we will study elaborately, but DNA is a genetic material which results in the carrying of genetic information. So when there is some differences in DNA copying, then two individuals are different from one another. So we can say that asexual reproduction has a little diversity. Why sexual reproduction has two parents? Because in sexual reproduction there are two parents, father and mother, and every offspring receives some character of father and some character of mother. Since different offspring receive different combination of characters of their parents, like father and mother, they show distinct differences among themselves as well as from their parents. The variation accumulate and pass on to more and more individuals with each generation. We will observe distinct variations when we see the people. Okay, and when you must be seeing your cousins, your many of the relatives, you must be observing that their parents look different from each other, their parents look different from each other. So these diversity are carried from one generation to another, and therefore no one resembles uh, equally from any of the generation, because there are some differences in any of the traits and characters. During sexual reproduction, variations are caused by four things. What four are? Chance separation of chromosomes during gamete formation. Maybe there is separation of chromosomes. Second is crossing over during meiosis. Third is chance coming together of chromosomes during fertilization. And fourth is mutations, that is alteration in the genetic material. So these four characters are responsible for variation in next one another generation. Now, who had discovered this? Why we are studying this? Some must have contributed in the field of genetics. The main contributor was Gregor John Mendel. He was known as father of genetics. So, Gregor John Mendel worked out the basic rules of such inheritance of traits more than a century ago. He studied the inheritance of contrasting characters, traits such as tallness, dwarfness of plants, round wrinkle form of seeds, axillary terminal position of flowers, yellow-green color of seeds, white-violet color of flowers, and so on. The work on the garden pea, that is the scientific botanical name is Pisum sativum. So the Gregor John Mendel discovered this genetically the hereditary information through a plant, and that plant was pea plant, Pisum sativum. He experimented on plant of various traits. But why he only chose pea plant? Because it was easy to grow, it had a short life cycle, and therefore it was possible to study number of generations. Garden pea had distinct, easily detectable, contrasting variants of features, like some plants were tall, some were dwarf, some had violet flowers, some had white flowers, some had round seeds, some had wrinkled seeds, and so on. They have number of traits which were contrasting to each other. So he just experimented on the same plant so that he could get how the transfer of characters takes place from one generation to another. So these number of traits was 7. And next point was though that the contrasting traits were existing in every generation, because plants had bisexual flowers and normally resorted to self-pollination. In these bisexual plants, artificial cross-fertilization could easily be achieved. Therefore, also he chose Pisum sativum. Each pea plant produced many seeds in one generation. This was one of the reason, and the last reason why he chose garden pea as for the genetic study was that they could easily be raised, maintained, and handled. There no much efforts were needed to grow a Pisces diamond plant. Therefore, he experimented on this. The traits which we will discuss are some contrasting traits which he experimented upon are plant size or height, first trait. Second is position of flower on the stem. Third is color of unripe pod. Fourth is shape of pod. Fifth is form of seed, shape or form of seed. Sixth is color of the seed. And seventh is the color of flower. These 7 traits he worked upon. While performing his crossbreeding experiment, Mendel took a number of precautions. He always focused on the inheritance of the specific traits under consideration and simply ignored others. For instance, in his cross, only one trait, that is size of plant, tall or dwarf, or color of flower, violet or white, was considered. He called such crosses as monohybrid cross, which is the cross. He crossed the traits of a plant. These were the traits were the contrasting characters and because he had took only one trait, the, the cross was called as monohybrid cross. Mendel also conducted crosses considering 2 or more contrasting characters, like one plant have 2 or different characters and another plant which he took will have also 2 or more different contrasting characters of the first plants. When he took two traits of a single plant, then this type of crosses which were made were dihybrid crosses or trihybrid crosses. Okay. Many short notes come on this chapter. This is an examination point of view. This chapter is very important. So we have discussed about the Mendels also. The questions also come like why Mendel considered pea plant for experiment only. What were the characters he took? Next, what other experimental techniques he followed for crossing for treating the plant? Next, write a short note on F1 generation, write a short note on dihybrid cross, trihybrid cross, monohybrid cross, test cross. So these could be asked like this. Another way of asking is they could give you a scenario and activity form that a plant is considered with this character and another character, like color option if they give that it has seeds of white color, one has seeds of red color. Then in F1 generation, which type of plants will be produced? What will be the production in F2 generation? So these types of questions could be asked from this chapter, children. So kindly study nicely in depth so that you can achieve more and more marks and have a good result. Okay. Now the crossbreeding we were discussing, that what is test cross, children? It is a cross between an organism of unknown genotype and a homozygous recessive organism. Now, children, this will be tough for you. I must express what is dominated recessive trait. Like children, if we will cross 2 plants of different contrasting character, like one plant is of tall and other plant is dwarf. If we cross and in first generation we obtain a tall plant, this first generation is called the F1 generation, and if tall comes in first generation in spite of crossing off, then tall is called the dominant character, and the dwarf character is called as recessive character. Means the character which expresses itself in first generation is dominant character trait, and the trait which is not able to express itself in first generation, it is called the recessive trait. Okay, now we will discuss a monohybrid cross, a breeding experiment dealing with a single character. Single character is a monohybrid cross. Like can you see in a diagram, this is monohybrid cross. Like a male pea plant is taken with round seeds, female pea plant is taken with wrinkled seed. Cross pollination occurs between them. The F1 generation we get with all round seeds. So this F1 generation have round seeds. Now this F1 generation is again self-pollinated. First we have done cross-pollination of 2 different plants. Now it is done in same plant, self-pollination. So in F2 generation, means in second generation, we obtain 3 round seeds and 1 wrinkled seeds. Okay. So such traits which were taken, only single traits were taken, and the crossing is done to say this is called a monohybrid cross. And you can see that F1 generation we obtain round seed. So round seed is a dominant character of the plant, and wrinkle seed are recessive trait of the plant. Now in the same way, Mendel's dihybrid cross, we can see two traits were taken, like a male plant with yellow and round colored male pea plant with round and yellow colored seeds. Two traits, round and yellow, means color and shape, both are taken. And was crossed with female pea plant with wrinkled and green colored seeds. The cross pollination was done, and in the first F1 generation, all plants with round and yellow colored seeds were obtained. This trait was then self-pollinated, and this results in F2 generation, in which four traits we obtain. For what we obtain? That in first combination was plants with round and yellow colored seeds. Second, plants with round and green colored seeds. Third, plants with wrinkled and yellow colored seeds. And fourth, which we obtained, was plants with wrinkled and green colored seeds. So these were the parental combination and new combination. This is the mixture of parental and new combinations which we get in F2 generation. So in this cross, we have took two type of traits in a single plant. So this is called a dihybrid cross. Okay, so whenever this type of short note or questions comes, you have to explain with the example, drawing a cross-like form in the table which we can see. So Mendel's, what he concluded is his conclusions on and findings by doing monohybrid and diploid cross. He concluded that in a monohybrid cross, only one of the two contrasting characters appeared in F1 generation. But in F2 generation, both the parental traits appeared in certain proportion. Means proportion is different, but they appear. In a dihybrid cross, when two contrasting pair of traits were considered simultaneously, only one parental combination appeared in F1 generation. But in F2 generation, raised by self-pollination, four combinations of trait appeared. These included two parental type traits and two new combinations. Okay. Mendel interpreted on basis of these conclusions. What he interpreted, he postulated that there are a pair of unit factors controlling each character in pea plant. One inherited from each plant. Mendel considered these factors as the carriers of hereditary information from one generation to another, that is, from parents to the offspring. At the time of reproduction, when gametes are formed, these factors segregate so that each gamete receives only one factor of each character. This is called the law of segregation. This what we discussed is the law of segregation, in which the characters are segregated and form a new trait, new character. Fertilization brings these two factors again together in the offsprings. Second, what he interpreted was, in F1 generation, only one character was expressed. Mendel called it as dominant character, which we discussed. The character which was not expressed was termed as recessive character. This phenomenon of appearance of only one of two contrasting characters in F1 generation is termed as dominance. Okay, it has dominated over the recessive one. Third, the characters are not lost even when they are not expressed. It was the third interpretation of it. The characters are not lost even if they are not expressed. They remain in the same. Fourth, when F1 offsprings were allowed to be self-pollinated, both the parental traits were expressed in definite proportion in F2 generation. If F1 offsprings were allowed to be self-pollinated, both the parental traits were expressed. So last interpretation he has done was, from the F2 generation of a dihybrid cross, Mendel postulated that inheritance of factors controlling a particular trait in an organism are independent of the other. This is called the law of independent assortment, like which we can say that while they are producing a different trait, they are not completely dependent on each other. They are completely independent. And inheritance, what is inherited from one generation to another, and the different traits are being produced, but this is the totally independent work. So this is called the law of independent assortment. Hence, at the time of reproduction, two pair of factors, each of the two traits in a dihybrid cross segregated independently during gamete formation and randomly formed combinations in the F2 generation. This is why new combinations appear along with parental combinations. That is why we are too different from our parents, from our grandparents, or maternal relatives, from our paternal relatives. Some resemblance are there, but we are not a clone of anyone. We are not being a human species. We have a physical traits similar to all the human species have. But like some features are different, some behavior patterns are different. So these are the variations. So student, today's the intro today was video was for about the introduction of what I have given about the genetics, about the hereditary information which we carry. In another video, we will elaborate this more and let you gain more knowledge about this hereditary information or genetic material. I hope you must have liked the video and please keep appreciating, please keep sharing among your friends and do comment to my videos if you like so. Thank you so much for watching. Keep learning and keep growing. Bye bye.